Hello, dear participant. We are in a SWAM course of Reproductive Animal Physiology. Module name is Reproductive System of Animals. Title: Reproductive Physiology, Reproductive Hormones, Oogenesis and Follicular Waves. Hi, I am Dr. Manish Kumar, Assistant Professor in the Department of Veterinary Physiology and Biochemistry, Faculty of Veterinary and Animal Science, Institute of Agricultural Sciences, RGSC, BHU. We will be continuing the reproductive system of the female, and in the last week of last class we have started with the introductory class of the female reproduction, and we talked there like what are the female reproductive organs are there, how their sexual differentiation in the male or female is taking place, and how the follicles grow, means how the follicles are there, primary follicles, secondary follicles and grafted follicles. So we'll begin with the new topic in a week 8 and lecture number 1. That overview of this topic is reproductive physiology, hormones involved in the female reproduction, oogenesis, follicular wave, and ovarian cycle and ovulation. Reproductive physiology of the animal. In a vertebrates, male and female reproduction involves interaction of internal environmental factor mediated by the hypothalamic pituitary gonadal axis. In mammals, there are two separate but coordinated cycles are there. One is ovarian cycle and second one is uterine cycle. Ovarian cycle, in which an oocytes mature in the ovary and ovulate to travel to the uterus. Ovaries perform the dual function of producing the ova, that is oogenesis, and secreting the female sex hormone, that is estrogen and progesterone. Uterine cycle, in which the uterine linings are being prepared for the fertilized egg to be implanted in the uterus. If the fertilization occurs, the cycles are interrupted while the female system adapts to nurture and protect the newly received or conceived embryo until it has developed the ability to live outside the maternal environment. Mammalian female continues her reproductive duties after birth by producing the milk for the young's nourishment. Thus, the female reproductive system is characterized by a complex cycles that are interrupted only by more complex changes, that is pregnancy. The ovarian and uterine cycle together constitute an estrous cycle. In most mammals, defined as the time from the one period of sexual receptivity to the next. So, these two cycles, that is ovarian cycle and uterine cycle, these together form a estrous cycle. And what is the estrous cycle? It is the time from period of one sexual receptivity to the next. Sexual receptivity is called as estrus or heat, which occurs periodically under the certain specified conditions, often at the time of ovulation. Ovarian cycles are very similar among the mammals, but uterine cycle differ in two versions. In the estrus type, if there is not a pregnancy, the uterine lining that has been prepared for the embryo is reabsorbed. In contrast, in some primate and humans have a menstrual cycle in which uterine lining slough off periodically, that is called as menstruation. The cycle begins at the end of each menstruation and occurs uniformly throughout the year. The duration of the estrus and menstrual cycle is species specific but variable within the species. The frequency of estrus cycle differs between the species. How frequently the estrus cycles will take place, it will be the different species of the animal. And On the basis of the estrus cycle, animal has been divided into group like continuously polyestrous or seasonally polyestrous. So, continuously polyestrous means cycle occurring uniformly throughout the year. For example, cattle, swine and rodents. Seasonally polyestrous means the animal will come into the estrus many times in a particular season only. Now, seasonally animal or seasonally breeders will be exhibited the estrus cycle as a day length. There will be short day breeder exhibit the estrus cycle as the day length decreases like deer, sheep and goat, and there will be long day breeder. They come into the estrus as the day length increases. Example, bears, hamster and horses. Now, seasonally monoestrus females, the most carnivores include the bears, dog, foxes, wolves are characterized having the single estrus cycle in a particular breeding season. In these animals, period of estrus is prolonged and lasts for several days in the seasonally monoestrus animal. Reproductive capabilities begins at puberty in a mammalian female, but reproductive potential of many primates, some rodents, whales, dog, rabbit, elephant and domestic livestock ceases gradually during the middle age, unlike the males. Primates with the menstrual cycle, this season is called as menopause due to the completion of the season of the menses. Ovarian cycle are similar in many ways among the vertebrates. Differences can be found in the type of ovulation, which type of ovulator that particular animals are there. They have been divided in a spontaneous ovulator. That means that ovulation is a regular process. It will take regularly whether copulation is taking place or not. Whereas reflex or induced ovulation, that means ovulation is induced by the stimulation of the sensory receptor in the vagina and cervix during the coitus. This either mechanically or by the semen component like cat, mink, ferret, rabbit, llama, alpaca and camel. These are reflex or induced ovulator animals. Hormones of the female reproduction. The ovary is the primary female sex organ that produces the two main steroid hormones. That ovarian hormones exert their action on the female genital tract and the mammary gland. The principal hormone associated with the ovarian cycling pregnancy parturition are estrogen, progesterone and gonadotrophin. Granulosa cells of the ovarian follicle, placenta, corpus luteum and adrenal cortex also produces the estrogen. Plant estrogen is also found in the legumes, that is clover and alfalfa, which causes the infertility in the animals. Now coming to the functions of estrogen. Generally, the principal function of estrogen is to cause the cellular proliferation and growth of the tissue related to the reproduction. So, estrogen is involved in the cellular proliferation and the growth of the tissue or organ which is involved in the reproduction. Action on the CNS of the estrogen is it promotes the female sex behavior and growth of the female reproductive tract. It induces the behavioral stress sign in the female, regulates the release of the FSH and LH from the anterior pituitary. Action on the uterus, it aids in the sperm transport by increasing the myometrial contraction. Estrogen priming promotes the growth of the endometrial gland necessary for maintenance of the zygote, that helps in the implantation of the zygote. Sensitizes the pregnant uterus to oxytocin at the time of parturition, thus aids in the uterine contraction along the oxytocin and PGF2 alpha for the expulsion of the fetus. Secondary sexual characteristics. Estradiol is responsible for the secondary sex characteristics of the female. It causes the growth of the duct system of the mammary gland, early union of epiphysis with the shafts of the long bones where the growth of the long bone ceases. It causes the deposition and distribution of the fat in the body. Its effect is probably associated with more specifically with the sex organ. Coming to the progesterone, progesterone is a sex hormone produced by the corpus luteum of the ovary, placenta and adrenal cortex. On the basis of the production of the progesterone, animals are divided into two groups, that is a placental dependent or corpus luteum dependent. CL dependent means animals are cow, goat and sow. So, corpus luteum virum is maintain the secretion of the progesterone throughout the gestation period, and it is responsible for the maintenance of the pregnancy. Whereas placental dependent animal, after a certain time, the placenta will secrete the progesterone and corpus luteum will regress and maintain the pregnancy. Function of the progesterone: it promotes the endometrial growth, it stimulates the secretory activity of the oviduct, endometrial gland and provide the nutrients for the developing embryo before implantation. Responsible for the lobular and alveolar growth in the mammary gland, prevention of contractility of the uterus during the pregnancy, regulation of the secretion of the gonadotropin, acts synergistically with the estrogen to induce the behavioral signs of stress during the pregnancy. Progesterone exhibits negative feedback mechanism, inhibits the FSH output and pulsatile LH release from the anterior pituitary. Gonadotropin, follicular stimulating hormone, luteinizing hormone, that is LH, are collectively referred to as a gonadotropin because of their role in stimulating the cells within the ovaries and the testes. FSH and LH are the hormones secreted by cells within the anterior pituitary. The main functions of FSH in the female is to promote the growth of the follicle, and LH is involved in the ovulation of the follicles. FSH and LH concentration exist in a plasma at a tonic or basal level. Tonic levels are increased by the estrogen and decreased by the progesterone. The release of FSH and LH from the anterior pituitary is controlled by the releasing hormone from the hypothalamus. Peptide factors secreted from the ovary are inhibin, activin, relaxin and oocytes maturation inhibitor. Inhibin, it produces the granulosa cells, it inhibits the FSH release. Activin, it stimulatory effect on the FSH release. Relaxin, it is a source is a corpus luteum, it dilates the pelvic structure, cervix and vagina prior to the parturition. Oocytes maturation inhibitor, the source is a granulosa cells of the primordial and developing follicles, maintains the oocytes at dictyate stage of the meiosis. Let us talk about the oogenesis, follicular wave and the ovarian cycle in the domestic animals. So, oogenesis is a formation of haploid ovum in an ovary by meiosis is called as oogenesis. Oogonia undergo the mitotic division until the final generation of the oogonia enters the phase of the meiosis 1. At this point, it is called as primary oocytes. This is under the influence of meiosis inhibiting factor. In domestic animals, except beech and queen, oogonia develops into the primary oocytes shortly before or after the birth. The nucleus of the primary oocytes enters the dictyate or resting stage of the meiotic prophase 1 and does not complete until the animal reaches the maturity. Resumptions of meiosis beyond the dictyate stage depend on the reproductive surge of the LH. After puberty, the rising level of FSH causes the follicle cells to develop. Contact between the oocytes and the granulosa cells prevents the maturation of oocytes beyond the dictyate stage through the oocytes maturation inhibitor produced by the granulosa cells. Oocytes maturation inhibitor concentration declines as the follicle matures. Ovulatory LH surge block the transfer of the oocytes maturation inhibitor from cumulus cells to the oocytes and allowing the meiosis to resume. During the last part of the fetal life, oogonia begins the early step in the first meiotic division but do not complete it, which is known as primary oocytes. Primary oocytes contain a diploid number of 2n replicated chromosomes which are gathered into the homologous pair. Primary oocytes remain in this state of the meiotic arrest until they are prepared for the ovulation. Before birth, each primary oocyte is surrounded by a single layer of flattened granulosa cells. Together, an oocytes and surrounding granulosa cells makes a primordial follicle. At birth, only fractions of the original primordial follicles remains, each containing a single primary oocyte capable of producing a single ovum. Most mammals, no new oocytes or follicles appears after birth. Follicles already present in the ovaries at birth serve as the reservoir from which all ova throughout the reproductive life of a female must be drawn, and only a small percentage mature and release the ova. The pool of the primary follicles present at birth give rise to an oogonia trickle of the developing follicles. Once it starts to develop, the follicle is destined for one of the two fates. One is either it reaches the maturity and ovulates, or it will degenerate. Of the initial pool of the follicle, most never ovulate but instead undergo the atresia at the some stage of the development. Formation of secondary oocytes and secondary follicle. The primary oocytes within the primary follicle is still a diploid cells that contains 2n double chromosome. A portion of the resting pool of the follicle start developing into the secondary, that is antral follicles. The number of developing follicles at any time is roughly proportional to the size of the pool. The development of the secondary follicle is characterized by the growth of the primary oocytes by expansion and differentiation of the surrounding cells layer. Oocytes enlargement is due to the build-up of the cytoplasmic or yolk material that will needed by the early embryo. The follicle next acquires the dependence on the gonadotropins, in part as a result of the differentiation of the cells surrounding the granulosa cells into the theca cells, which markedly increases the estrogen production by the follicular cells. Just before the ovulation, the primary oocytes whose nucleus has been in the meiotic arrest been for years complete the first meiotic division just few hours before ovulation, except in the bitch and mares in which ovulate the primary oocytes. In association with the extended period of estrus and completion of the first meiotic division and extrusion of first polar body occurs after the ovulation. This division yields two daughter cells, each receiving the haploid set of the n doubled chromosome. However, almost all the cytoplasm remains with one of the daughter cells, now called the secondary oocytes, which is destined to become the ovum. The chromosome of the other daughter cells together with the small share of the cytoplasm is called as first polar body. In this way, the ovum to be loses its half of its chromosome to form the haploid gametes, but retains all its nutrient-rich cytoplasm. The second day, oocytes enters into the second meiosis. It completes when the spermatozoa penetrates the zona pellucida to activate the oocytes to trigger the second meiotic division. Secondary oocytes that are not fertilized never complete this final division. During this division, the half set of the chromosome along with a thin layer of the cytoplasm is extruded as a secondary polar body. The other half set of the n unpaired chromosome remains behind in what is now the mature ovum. These 1n material chromosome unite with 1n paternal chromosome of the penetrating sperm to complete the fertilization. The first polar body has not already degenerated if it to undergo the second meiotic division. At the same time, the fertilized secondary oocytes is dividing its chromosome and form the two polar bodies. Now coming to the follicular waves, there are 3 to 6 follicular waves are initiated to grow together under the influence of increasing level of FSH, which is described as follicular wave. The first wave of the follicle that develops during the mid-luteal phase reaches the antral stage and then regress, followed by growth of the second wave of the follicle. If the CL regression begins at the time one of these follicles become dominant and these follicles reaches the ovulatory stage, rest of the growing follicles regresses and become the atic follicle. The dominant follicle produces inhibin or estradiol, which inhibits the FSH, thereby growth of further antral follicles is blocked. In sheep, cattle, horse, 2 to 3 follicular waves occurs during the luteal phase, with the last wave ending in the ovulation. In monotocous animal, at each heat period, normally one follicles develop more rapidly than the other and releases one ovum. Rest of the growing follicle regress or they form the atretic follicles. In polyestrous animal, more than one follicles grow and more than one follicles will ovulate and fertilization will takes place. Following the ovulation, the follicular antrum is filled with blood and lymph that forms the corpus luteum hemorrhagicum. This is gradually converted to the corpus luteum. After each estrous cycle, CL decreases in a size and finally leaves a scar that is called as corpus luteum albicans if pregnancy is not established. Coming to the ovarian cycle, the shape, size and functionality of the ovaries vary both with the species of the animal as well as with the stage of the estrous cycle. In the cattle, it is ovary is almost almond shape. Horse, it is a bean shape. Sow and birds, the ovary resembles a cluster of the grapes. In the chicken, only left ovary and oviducts are normally functional, whereas if left ovary and oviduct is removed or any diseased condition is there, so right ovary and oviduct will be functioning. After the onset of puberty, mammalian ovary alternates between the two phases, that is a follicular phase and the luteal phase. Follicular phase means in the ovary, the structure you will find is follicles. There will be number of follicles, but one growing follicles will be there. The follicular hormones also govern the development of the reproductive tract and facilitate the sexual receptivity. Luteal phase, in which on ovary the structure corpus luteum will be available, which is a source of the progesterone, and progesterone is important for the maintenance of pregnancy. In estrus mammal, the follicular phase is comparatively small, usually encompassing not more than 20%, whereas luteal phase is encompassing around 80% of the total period. When the reproductive cycle begins, the selected follicles within the ovary are influenced by the hormones and proceed through the growth and maturity, followed by the ovulation and development and regresses in the corpus luteum. Puberty is defined as the beginning of the reproductive life, which in the female is usually marked by beginning of the ovarian activity. The formation of the Graafian follicle from growing follicle is a hormone dependent, begins at puberty when tonic level of LH and FSH begins to rise and fall with the each estrous cycle. The interstitial cell begins to surround the basement membrane of the granulosa cells to form the theca, which differentiate into the theca interna and theca externa. As the thecal cells are formed around the follicle, a capillary bed develops among them. These thecal capillaries increases in size and are concentrated in a theca interna close to the basement membrane that separates the theca interna cells from the granulosa cells. LH receptor form on the cells of the theca interna and receptors of the FSH and estrogen forms on the granulosa cells. During the hormone dependent stage, under the influence of LH, androgens are produced by the cells of the theca interna. The androgen diffuses from the theca interna to the granulosa cells. Under the influence of the FSH, these granulosa cells convert the androgen into the estrogen. The estrogen produced causes the growth and division of the granulosa cells, and together with the FSH causes the granulosa cells to produce secretions that affect the separation of the granulosa cells and formation of the space filled with the fluid called the antrum. Also, FSH stimulates the formation of LH receptor on the granulosa cells. A surge of LH output, pre-ovulatory surge, occurs 24 hours before ovulation. In addition to its role in ovulation and formation of the corpus luteum, LH surge causes the reduction in the number of FSH receptor on the granulosa cells, so that the output of the estrogen by the granulosa cells is decreased. Two gonadotrophin theory says that according to this, luteinizing hormone stimulates the thecal cells to produce the androgen, and the follicular stimulating hormone stimulates the granulosa cells to produce the estrogen from the androgen. Here you can see this is the theca interna cells, this is the capillary network, and this is the granulosa cells, and this is the basement membrane. Coming to the ovulation. Although ovarian cycles are similar in many ways among the vertebrates, differences can be found in the type of ovulation. The most animals are spontaneous ovulator, ovulate with the regular frequency, and second one is a reflex or induced ovulator. That means they will be ovulate only the copulation is taking place, or stimulation of the cervix or vagina is taking place either by the copulation or by the seminal components. Ovulation. The selection of the follicles for the ovulation seems to occur primarily by the chance. It is usually associated with the largest actively growing follicle present when the previous CL is regressed. Follicles continue to grow and develop during the all phase of the ovarian cycle, with some impairment during the luteal phase, and the LH surge is necessary for the ovulation to occur. Follicles close to full development but without adequate LH receptor do not ovulate in response to LH surge and becomes atretic. Corpus luteum formation and regression. The formation of the CL involves the luteinization of the granulosa cells, by which the granulosa cells converted from the estrogen secretion to the progesterone secretion. The process in which it initiated by the pre-ovulatory LH surge. The cavity of the ruptured follicles and fibrin clot within serve as the framework on which the granulosa cells develop. Blood vessels from the theca externa invade the developing CL and it becomes the vascularized. Maintenance of the CL is provided by the LH derived from the LH surge and by the basal circulating level of LH. In the sheep, prolactin, a gonadotrophin hormone for some species, is required to maintain the CL in addition to the LH. The uterus play a major role in controlling the lifespan of the CL in non-pregnant mares, cow, sow, ewes and doe, but not active in CL regression in the bitch and queen. PGF2-alpha is released by the non-pregnant uterus about 14 days after the ovulation, and it is considered to be the natural luteolytic substances. To ensure that enough PGF2-alpha reach to the corpus luteum for the regression of the corpus luteum, that veins and artery are anastomos, means they are passing side by side and it is being transported to the corpus luteum. Survival of PGF2-alpha in general circulation is more important in the sow and mare. So in this we have seen that how that ovulation is taking place, how that stress in the animal is there, what type of animals are there, stress, monostress, polystress, seasonal breeder, seasonal polystress, seasonal monostress animal, and how the ovulation is taking place. So this we are completing this topic. Thank you.